All right, well, I'll sit in this. All right. So like I said, what we're doing in chapter 14 is, we got this idea of heredity and how genes are inherited and dominant alleles. And, we've looked at chromosomes under the microscope, and we've even talked about them, from way back in, chapter 3, whichever chapter, the ones where we talked about organic molecules, about nucleic acids. And we even discussed them molecularly back then, if you remember right, when we were talking about the monomers and the polymers. Remember, we talked about what a nucleotide was, and that there were adenine and guanine, and it was different in RNA and DNA from one of those bases, right? Thymine and uracil and all that. So what I like about this course and the way it's all set up is we're kind of going full circle and coming back at that again, because at the very end of a general biology course, it's always about heredity, because that is one of the most important and one of the cutting edge kind of things that's going on in society today. So it's really important that we kind of talk a lot about that. Everything else is important, photosynthesis and respiration, but when it comes right down to it, we're pretty well informed about how those things work. It's heredity that really holds not only a lot of unknowns to us, but a lot of potential, a lot of ideas. I mean, we're talking about the actual recipe to life. So as I said, it's been a while that we've been studying these things. First of all, trying to figure out what the heck was heredity, and what was given from one parent to the next to make them look like that. And so, of course, we were getting better at making microscopes, and so we were looking through a microscope, maybe similar to like this that we saw in class the other day with the onion cells, right? And the fact that the chromosomes were being stained and standing out very well allowed us to not only see them, but kind of figure out what they were doing. So Miescher in 1869, he looks in the nucleus, and he, like I said, he might have been actually, I could go back and research it, but he might have been actually watching cells as they went through mitosis. So he kind of did a common sense kind of thing, I guess. So, well, if genetic material is the key to the cell, if it is the information about the cell, if it's everything the cell needs to stay alive, if that's what we're postulating, then it makes sense you're going to copy when you copy a cell, right? All right, so then Fuelgen 1900s comes up with a stain, that stain that you see in the picture. So now it's going to be really easy to, if you stain them properly, to watch them move around during mitosis, to actually start to get an idea of their structure. And so we're starting to have what you would probably call circumstantial evidence at that time, right? This is probably where the genetic molecule is. It's probably in the nucleus, and it's probably these little purple wormy things. Sorry, I'm opening the door to the room. It's getting a little bit warm. These little purpley things that we've been seeing under the microscope. So Griffith, if you remember, now I'm going to come back to Hershey and Chase in a second. I noticed the other day my slides were out of order, so I need to actually switch this slide with the next one, but I'll come back to that one in a moment. Griffith, if you remember, was the fellow who was working with the Streptococcus organism, the organism that causes pneumonia. And we said there were two strains. There was an S strain and an R strain. And the S strain had this, it's in orange there, had this sort of mucus covering around the two cells. And if you had taken my Bio 2 class, you learned that a lot of prokaryotes that are pathogenic, disease-causing, they have this capsule around them. It hides them from the immune system. It protects them from the immune system. Your immune system knows that things, we talked about this, I think we were talking about cell membrane structure. Your immune system only knows cells are foreign or not supposed to be there if it can feel them, essentially, right? So without that capsule, it can't. And then those organisms kill the mouse, and without the immune system being able to stop that, right? The living R, which is another strain, that's kind of the word we like breeds in dogs. We use strains among prokaryotic cells. It's still the same species, but since it doesn't have a capsule, the mouse's immune system does a good job and destroys it, and maybe the mouse gets sick, but he doesn't die from it. Griffith continues on with his work by making sure that this is what we call controls. He's got a pathogenic control, which basically says injecting the mouse with living S kills it. Non-pathogenic says injecting the mouse with living R doesn't kill it. The heat-killed S is also a non-pathogenic control, because what that means, non-pathogenic, is not caused by the critter. All right, so in this particular case, it's a non-pathogenic control, because even though those S cells usually would kill the mouse, they have to be alive to do it, you see. It's not the mouse's immune system over-responding. It's not like those folks that are extremely sensitive to peanut butter, right? Like on airplanes now, they can't serve peanut butter because if you're really super allergic to it, just the smell of it can send you into anaphylactic shock. And in anaphylactic shock, your the air vessels in your lungs spaz and tighten up, and you literally kind of suffocate. All right, so with this heat-killed S, we're going to determine that it really is the organism that's doing it. Now, and the last one is really kind of where he got this, or really where he sort of lended us evidence about what genes can do and how they store information. He mixed the heat-killed S and the living R together, and the mouse dies. Now, neither one of those should have killed the mouse, and when he cut open the mouse, he noticed that inside were living S, which he did not inject. What had happened, of course, is the information in the dead S, as I said the other day in class, just because you're dead doesn't mean your DNA has disappeared. So that information is still there, and the living R cells, because you can't kill something if you're dead, so the heat-killed S cells are out of the equation as far as killing the mouse, but the living R cells can engage in something that we now know bacteria do. It's called bacterial transformation. It's one of the ways that bacteria increase their genetic diversity. Remember, they don't go through meiosis, so they don't shuffle up genes the way we do. They do it in a variety of ways involving sometimes viruses, just reproducing however many they make, and sometimes they can take up DNA from the environment. I mentioned in class even different species DNA. Now, I don't know exactly how far that goes. I mean, is that probably different prokaryotic species, but I'm not sure it can take up the DNA of you and become a human bacterial cell. I don't think that would happen like that. So the living R took up the DNA from the heat-killed S. So when the heat-killed S died and lysed and all the membranes broke apart, that those chunks of DNA would have all been scattered apart, scattering throughout the environment there inside the mouse. The living R would have taken that up, began to read that DNA, remember it's information, and would have been transformed or churned into living S. Simply, it would have learned how to make that gel or that capsule around it, so the immune system of the mouse couldn't see it, and the mouse dies. So again, the importance of Griffith's work is the fact that he showed that this molecule, this genetic molecule, this whatever this thing that we were looking at, under the cells, that there is a thing that can happen in certain cells, like we can't do this, but bacterial cells, where they can actually take up some of this DNA molecule and change who they are. You see what I mean? Go from a living R to a living S. And if you're changing who you are, you're changing your genetics. As I said, this is something that bacterial cells, since Griffith's time, have been known to do, or have been shown to do in a variety of other cells. The bacterial transformation is something that other bacteria do, and that literally means they take up DNA from the environment that changes who they are. All right? So that's interesting, because remember, DNA is an interesting molecule because it stores information. It's not like water. Water doesn't store information, it's just H2O. In the 1950s, so now we're getting close, because the Watson and Crick, the guys at the bottom, they were the ones who got the Nobel Prize, you can see in the picture there, for putting all of this together. That's obviously a photo op picture of them at the bottom, where this guy's pointing to it, this guy's looking at it. These are Watson and Crick, and Crick is on the right. No, Watson's on the right. Watson's the British fellow. He's on the right. Crick is on the left. Very different, odd folks. But they built a model of the DNA, and this is, like I said, probably a Photoshop. But just prior to that, and so at this time, it's like a beehive of work on this molecule, man. There's tons of people, like the woman you see in the picture there, Rosalind Franklin. She did work where they were X-raying a DNA molecule. Now, if you look just to the right, you can see the picture of the X-rayed molecule. If you're kind of going, well, how's that DNA? Remember, DNA is a double helix, like two springs wound together. I don't really have a model to show you, but imagine you were looking not at the side of the DNA molecule with the twisted ladder, but down the ladder, down the twisted ladder. That's what you would see there. So this X-ray shot is if they were looking along the vertical axis of the DNA molecule. And the little blips that you see there, the little blips and the little X-ray exposure things that you see are indicative of hitting things, hitting bases, hitting phosphates. And they could tell, in some cases, what it is they were hitting. And every time they took a picture of DNA, and this is interesting, every time that Wilkins and Franklin, okay, Franklin was her partner. No, she's Rosalind Franklin. Maurice Wilkins was her partner. Every time they took a picture of DNA, those blips came out in the same place. Now, I'm not an X-ray tech, but you don't need to be an X-ray tech to figure out that if what we're doing is taking a picture of molecular structure, that if those blips are in the same place, that means there's a regular arrangement to those blips, whatever those blips might be. Could be bases or phosphates or those ribosugars or deoxyribosugars we were talking about. You see what they're doing here, obviously, is they're learning what the pieces are, and now they're kind of learning how to put them together. It's almost like a really big, giant tinker toy thing to do. And that's, if you look at the picture of Watson and Crick there, that's what they're doing. They're down building this thing. You know, they had computers, but not computer-aided design. So they're literally putting this thing together with sticks and glue and paste and gosh, you know, whatever, and then trying to see if it fits the data they have based on the way they've constructed it. Let me jump back to Hershey and Chase, though, for just a second, because I mentioned something in class the other day. There's the work. About initially the DNA or the genetic material being thought of, you know, is it a protein? And the reason why that was because at the time we only knew about carbs and lipids and proteins. We didn't know about nucleic acids at the time. Remember those 4 organic molecules we talked about? Well, since a protein had code to it, the amino acid sequence, we were kind of like, oh, code. Code makes sense. Code contains information. Code can make things look certain ways, you know. So we're thinking, that's a good idea for the genetic molecule. Maybe it's a protein. So what Hershey and Chase did was demonstrated quite clearly. In addition to the protein enzyme, remember I told you they took a protein enzyme and put it on the cell, and they noticed that it digested the nuclear contents. So in other words, they disappeared. Not all of it did, by the way, which we'll talk about in subsequent chapters, why all of the protein didn't go away. But most of it did. So they said, that probably means this genetic material is not a protein, since protein enzymes got rid of it. So Hershey and Chase built upon that in their experiment. What that weird-looking spacecraft-looking thing is called a bacteriophage. It's a virus is what it is. And viruses are things that are kind of not alive but are somewhat alive. Biologists who kind of go to the non-alive side, which is kind of where I am on the discussion, tell you that viruses can't reproduce by themselves. Viruses need a host cell. You see the thing in the brown down there? That's a host cell. That virus is going to attack that cell, and it's going to inject something into it. And what it's going to inject into it is going to make that cell make more viruses. Now, those viruses, they don't metabolize. They certainly evolve. They are organized, okay? Response, I'm sure they probably do, but you know, they don't have all the characteristics left. They don't metabolize. They don't reproduce in a traditional reproductive way. All right, they use a host cell to make copies of themselves, and then what they do, that stuff that's being injected into the cell in both those pictures, it's either brown or red in the other one, that stuff is going to wire the cell. It's going to program the cell. It's going to take over the cell's machinery, protein machinery, its ribosomes. It's going to do it by attacking that cell's genetic information. And it's going to tell that cell, make viral particles, make lots of viral particles. And not only make viral particles, make the legs and the heads, and viruses all have different shapes. This is just the way a bacteriophage looks. All right, so you understand what we got going on with, you know, how this all works with the bacteriophage. All right, so what we do is we take one bacteriophage, and we can manipulate these things, and we cram the head up there, you see on the one on the left, with radioactive protein. There is no sulfur element. Notice the radioactive protein is an isotope of sulfur, right? There is no sulfur in DNA, but there is in proteins. All right, so follow me along how the experiment is wonderful. It's brilliantly simple. Okay, so they take the one on the left, and they stick the brown, and the brown stuff is protein, right? And they take that protein and they radioactively label it. All right, and then they have that bacteriophage act as the vector and inject that radioactive protein into the cell. They say it's cold DNA because they're looking for the other type, the other side of the picture that you see there in just a minute. So then they put them in a blender and they centrifuge all that up, and they find that there's no radioactive protein inside any of the cells. They let it grow for a while. Now remember what I said, what viruses do, they inject something into the cell makes more viruses, and then the cell bursts, and they go out and attack more cells. So after a period of time, the blending and the centrifuging, by the way, is to separate the pieces. They don't find that radioactive protein anymore, so it's not being copied. See what I'm saying? It's not being copied. Miescher's work in the nucleus, Fulgen's work in the nucleus, everybody looking in the nucleus going, something's being copied. Quite clearly, we know the way a virus works. It works by injecting genetic material into a cell. So is it a protein? Doesn't appear to be. Now, let's turn around and go the other way. The hot DNA. We inject, or those phages inject the stuff in red, which has radioactive phosphorus. Now, DNA does have phosphorus, remember. It's the phosphate groups that hold the links together. So there's no sulfur in DNA, but there is in proteins. Now, there might be phosphorus in proteins, okay, but there's no sulfur in DNA. You see how they separate those two off? So they take the radioactive DNA, radioactive because the phosphorus in it has been made radioactive, and they have sensors to detect that. And they do the same thing, inject it into a bacteria blender and blend and centrifuge, separate it all out, and guess what? Cells, tons of cells have that radioactive material in it. And since it's radioactive DNA, we see, there we go, DNA is the hereditary molecule. Now, the discovery of DNA, or at least its complete function with structure with Watson Crick, is going to happen soon after this. But you see how this experiment is kind of leading to this idea, okay, guys, we're getting close. This is the molecule. We can forget the protein hypothesis, what they did. You know, in scientific method, you refute the hypothesis, boom, it's gone. Support another one. Looks like it's nucleic acids, folks. It looks like that's our hereditary molecule, so we're going to run with that. So that was the significance of their work, definitely showing us that it was not a protein. Now we were on the right track. We were getting warm to knowing exactly what it was. So then Wilkins and Franklin come along and did their X-ray work and showed us how maybe to put the pieces together. All right, so we know that's the molecule it is. Now we got folks who are learning what it's made of. Chargaff, for example, figured this out. Figured what out, you might be saying. Chargaff figured, here's how he did it. Whenever he looked at a piece of DNA, Chargaff was a researcher, he found that the concentration, remember I said if you put brackets around things in chemistry, it means concentration. He found that the concentration of adenines, that's supposed to be a bracket, always in a piece of DNA, no matter what, from whom, from where, the concentration of adenines always equals thymines, and the same thing for the guanines and the cytosines. Now, you remember back in that earlier chapter that I referenced earlier in the lecture, we looked at the molecular structure of nucleic acids, and I told you at the time that they always bind A to T and G to C. So this becomes known as Chargaff's rule. Chargaff's rule is really important. Now, Chargaff, of course, the important part, in addition to the rule, is the fact that he helped discover these molecules and how they bind together. Well, the important part about all that is, number one, now we could see what the bases were as we went up the ladder, and we might notice that they changed, and we might notice that they might be different in different molecules, and we're like, ooh, the code is changing. That's cool. And especially if those different DNA molecules came from different critters, we're starting to put together, oh, is the code what they're like? And the other thing that Chargaff really did, Chargaff's rule, is if you know one side of the DNA, if you know the sequence of letters, if you know the code, you know the other side. It's always A to T and G to C. And that's how he did that. So we could even do a percentage. Let's try this out. Let's say I take a piece of DNA and I analyze it for adenine, the concentration of adenine. And we'll do it in percentages. And I say, all right, the concentration of adenine for this sample piece of DNA is 20, should I do that right? 20%. So if I look at a piece of DNA, 20% of the bases are the A. From that one piece of information, I can tell you the concentration of all three other bases. The thymine one is easy, right? What's the concentration of thymine? If adenine is 20%, what's thymine going to be? Yeah. All right, so how about guanine and cytosine? How would you do that? Between the two of them, yeah. Right? So what's the percentage of guanine? 40%. Are we doing that right? Wait. 20 and 20 is 40, leaves 60. Wait a minute, hold on. If it's 20% adenine, I'm sorry, Ryan, if it's 20% adenine, then you said it's 20% thymine, and that is correct, because A to T. 100% is what it totals up to, right? 100% of all the bases is all the bases that are there. These two add up to 40%. So 40% of the molecule is either adenine or thymine. So the other what? Other what percentage is guanine and cytosine? Yeah, each one, right? 60% between the two of them, and of course it's always guanine to cytosine, so it's 30 for each. Isn't that neat? So if you're looking at the, I mean, you don't know the order of the bases, but if you want to know what bases are in there, all you need to really do is figure out how much there is for one of them. Pick any one, and then use what we just did, and you can figure out all the other ones. All right, now we're ready to get on to Watson and Crick. As I said, they did this basically with sticks and glue and put it together, but we've got much better ways of showing what they finally came up with the idea. And they were awarded the Nobel Prize, I think, along with Chargaff, if I remember right. In 1952, I think they were awarded the prize in 1953, if I remember correctly. I'm not really sure, but it's early 50s. And they were awarded the Nobel Prize for this, right? They were the ones. Now, they weren't necessarily the researchers that were doing the grunt work of figuring out the sugars and the thymines and the binding and the things that Chargaff were doing and the thing that Wilkins and Franklin were doing with their X-ray diffraction. But what they did is they compiled all this data together and built these models, like I showed you, with the information they had, and then tested them to see if it made sense. In other words, did it seem to fit with everything else we know that this molecule can do? And here's what they came up with. First of all, they came up with a double helix. They decided the molecule was two twisting structures. Like a helix is a spring. So think of two springs twisting around one another. If you jump into the middle picture, I'm going to kind of move back and forth. If you jump into the middle picture, then those two springs were these things right here. They're the phosphates and the sugars that bind them together. It's the thing, it's the place, it's the area where monomers become polymers. Remember, that's how they get linked together. So if we think about DNA as being a ladder, then these might be like the sides of the ladder, right? The rails of the ladder. I know you've heard me say this before because I'm sure I said it back in the earlier chapters. And adenine binds to thymine, and those are the bases that fit across, all right? There's the T binding to an A, and it has to do with hydrogen bonds. It's actually very simple. Guanine and cytosine have 3 places for hydrogen bonds. That's why they stick together. Adenine and thymine only have 2. Okay, those numbers that you see there, that was the work of Wilkins and Franklin. Whenever they took an X-ray of any DNA from any critter, they found these blips on the X-ray. They found these structures, just like a doctor took an X-ray of your ankle, you could see the bones and so on and so forth. But they found them at regular dimensions and regular intervals, and it was always the same in each DNA. So they measured those things, and Watson and Crick used the measurements from the X-ray to figure out, for example, that the molecule is 1 nanometer, half of it. It's actually, the molecule is 2 nanometers wide. I don't know why they always go with that. Half of it is 1 nanometer. Well, then the molecule is 2 nanometers wide, right? The 3 point, well, no, let me do the 0.34, this one down here. There was another thing on the X-ray film that kept popping up at this distance. What they were hitting with the X-rays was the bases. And so the reasons why those bases were, the reason why those blips were certain distances apart, always the same, as a matter of fact, 0.34 nanometers, is because that's where the bases were. So that told Watson and Crick, okay, we know how wide the ladder are. R is whatever. They're twisted around each other. The bases that Chargaff has helped us figure out are adenines and thymines. They're going to be arranged, and the rungs of the ladder are going to be 0.34 nanometers apart. Every 0.34 nanometers is another base pair. There's another T to an A to a T. Matter of fact, that's how they measure genes anymore. We measure an X number of base pairs. Like a gene X might be 40 base pairs long. So that would be an adenine and a thymine would be one base pair. And finally, the 3.4. The molecule twists. All right, that's why it's a helix. And it makes a small twist and a large twist. And it does that small twist and that large twist in a space of 10 base pairs. So it takes 10 base pairs to go through the small twist and the large twist. And so if you look down below and you multiply this number, which is the distance between base pairs, you'll notice that it gives you that number, doesn't it? I love that story, man. Isn't that something that's like little pieces of the onion coming off a little tiny bit at a time? Just a little bit. Imagine all the people working on this, and you know, at the very end is the space filling model, if you wonder what that is. But you can imagine all the folks working on this and just, you know, getting together just little pieces and building it up. Must be. There's a is it Watson? Yeah, Watson wrote a letter to his son soon after they discovered what the structure was, and he draws it in the letter, just very, very roughly, like what you see on the left. I think that letter just recently went for sale at some auction for, you know, gosh knows how much money. But that would be it had it online. It was cool to read. He's just kind of, dear son, kind of, I think we've got it, and he kind of draws it out. I'm like, oh my gosh, look at that. That is the DNA molecule. Okay, before I leave this slide, I do want to talk about what we mean by being anti-parallel. It's important. It has to do with chemistry. It's important not so much from the chemistry part, but how it affects when we go on to copy DNA later on, when we go on to replicate it, right, which we know obviously happens during mitosis and meiosis. What we mean by being anti-parallel, if you remember back in the early chapters when we looked at glucose, I said that we would number the carbons. Number 1 carbon, number 2 carbon, number 3. The little apostrophe, is that what that thing is? We don't call it apostrophe, we call it prime. So we have a 1' carbon, a 2' carbon, a 3' carbon, etc., right? What we mean by a 5' N, 3' N, 3' N, yeah, and 5' N on the other one is the way the two ladders are hooked up. On one of the ladders, the top rung, it's the number 5 carbon that is exposed. The number 5 carbon would actually be that one right. Come on, there we go. This one right here. That's the last carbon in the chain as you move up the chain. If you get down below and you look, then remember there's a carbon right there, even though it's not written. That's the number 3 carbon. That's the number 1, the number 2, the number 3, the number 4, the number 5, okay? So if we look on the other one, you'll notice that it's different. On the other strand, the 3' is up, and the 5' carbon is down. All right, that's what we mean by anti-parallel. So imagine if you took a ladder and you cut it right down the middle, and then you flipped one end. Now, that's the chemical structure, that's the chemistry that's allowed to be building that molecule. And as I've said before, biological living organisms are really nothing more than a fancy chemistry thing. So this is what physics allows. This is what chemistry allows. This is what chemistry says. This is how these things can bond together. As I said, I'll come back to this in a couple of slides, but this has very important implications for living organisms when they try to copy DNA, all right? So you'll see those terms again, 5' and 3', and I'll talk a little more about anti-parallel here in just a second, slide after next, actually. You see, because what we're going into is a couple of guys named Messelson and Stahl. They were trying to figure out how DNA replicated, how we made copies of DNA. So they had 3 hypotheses that you see over on the right-hand side. For example, DNA could replicate itself conservatively, and the pictures there are great, right? Conservatively means the original strand of DNA. There's 2 strands together, the 2 helices, and they haven't curled them up. They straighten them out for you there, obviously. But what is done is you make a copy. Like a conservative copy would be like if you went to a copy machine and made a copy of a piece of paper, like somebody's notes, right? Because you would have the copy and they would have the original. You wouldn't have the machine doesn't work in such a way where you get part of the original on your page and part of the copy. So it lifts some of the letters off the original, maybe half of the C, let's say, sticks it on your page, and then adds the other half of the C. No, that's not the way a copier works. A copier works simply by making a copy of something, right? Makes an exact copy. Okay, that's one way. That could be. These are hypotheses. This is how science works, right? The other one is called semi-conservative. And in semi-conservative, the way it works is you take the original and you split it in half. Now, sort of if I use the analogy for a copier and conservative, semi-conservative to me would be tracing. Like when I was a kid, if we had to hand in book reports and we wanted to do an illustration, before we were concerned about things like copyright, apparently, I can't draw for crap. So I would trace stuff. I would get that tracing paper, that onion skin paper, and I'd trace over it, and that would be my submission for my report. So the thing about semi-conservative is tracing. And we'll get back in a second as which one it is and how we knew. The other one is called dispersive, and dispersive basically means that the way you make a copy of a DNA is you take the two original gray strands and you chop them all up into pieces, and then you arrange them all back together, and the new and the old are all mixed in. Well, parsimony, which is a science idea or an idea that you ought to keep things simple, that the simplest explanation is probably the best one. Parsimony was probably one of the reasons, and they also did tests, that you would think that this last one, that's probably not it. And you're asking for trouble, right? Can you imagine if you take a piece of DNA, say 1,000 base pairs long, and you chop it up into pieces, and then you make new pieces of those copies, and then you try to rearrange those pieces back the way they were, and the order of the letters is important, you're going to mess something up. That'd be like me trying to freehand draw something if I was going back to my tracing example. Now, the way they figured out the way it was once again one of my favorite things. They made these things glow under a fluorescent microscope. So what they did is they took the DNA bands, the original ones that were in the gray, and they put them in and they allowed them to go through this replication. And they added other bases, because if you look down on the first one, we got 2 strands of DNA. We're saying we'll make a copy and now have 4. So we had to get the new bases from somewhere, the new A's and T's, to match up with the other ones. All right, so they radioactively labeled those. And when they did, they found out that it was the semi-conservative way, the way you might think, and if you think about it for a second, you probably can think why. It was done semi-conservatively. That's how DNA replicates. So between those 3, maybe we can even throw out dispersive, because we were just like, wow, that really seems like a way to make a mess. Why would you think semi-conservative might be favored in natural selection and Mother Nature over conservative? I mean, consider to be conservative replication, something's going to have to sit there and take the sequence of letters on the gray strands and assemble them into new strands identical in the blue strands. So something would have to do that, some machinery. So to me, conservative is a little like dispersive in the sense that you're introducing a lot of chance for error. Wouldn't it make more sense, and especially if you want to keep the DNA saying the same stuff? DNA does mutate and changes, and replication isn't always 100%, okay? But doesn't it make sense that the tracing one, the semi-conservative, is probably your best bet of getting a good, accurate copy of the DNA? If you split the gray, if you split the ladder in half, remember the bases are sticking out, and then you just fill in the other side of the ladder fit with Chargaff's rule, you're probably going to minimize your chance of making mistakes, right? What do you think about that? Does that make sense? So I guess in order to have conservative, we would have a DNA Xerox machine. That doesn't make any sense. All right, so let's keep that in mind, that the replication occurs semi-conservatively. What I mean by that is now I'm going to show you how it happens. And the first thing you got to know how it happens is what it's going to do to make a copy of a chromosome is it's going to split this molecule in half. All right. This is one side of the ladder. This is the other side. We're going to split it in half, use that as a base, and add the new bases to it. But it's not quite as simple as doing that. And the reason why it's not quite as simple as doing that goes back to that 3 and that 5 prime. You notice, okay, what we got here is we're copying a piece of DNA from left to right across the screen. The original piece of DNA is way over here on the right, and it's still coiled up. Here's the original piece. So we're moving left to right, and as you notice, all this stuff that we're going to talk about here in a minute, we've split the DNA, and then we're adding the other side. So we have, you know, the original strand is here from this piece of DNA. The original strand is here from that piece of DNA, and these are the new strands that are being added. And eventually we'll have 2 pieces of DNA, okay? Semi-conservative replication. We're keeping one of the old strands, keeping it the same, and we're adding a new strand and just following Chargaff's rules. All of those yellows and green little bases you see bonding together there, those are all going to go A to T, G to C, unless some screw-up occurs. Okay? Let's go back to the beginning now. Let's talk about all these things and their job. Helicase. Helicase is an enzyme. Helicase unzips the DNA molecule. Helicase would be like a boat moving through the water and pushing away, say, some floating weeds. That's its job. Its job is to go in there and break those bases apart. Now, if you were to take something coiled up, like a piece of DNA, and pull the strands apart, you would eventually create one heck of a mess down here, wouldn't you? Think about if you've ever tried to untangle the Christmas lights by just pulling the strands. It doesn't work that way. So what this little thing does, top isomerase, this is really pretty cool, is it breaks the DNA molecule. Every time tension increases in that twisting, it breaks it and reattaches it untwisted. So it breaks it, relieves the tension, and puts it back together again. It does it at the phosphate groups. It's kind of neat, right? So every time you're getting that kinking up, top isomerase enzyme goes in and goes blip, and undoes that. It's sort of like when you have to finally unwind the hose. You know, it's another, you grab the garden hose on the thing and run out into the yard, and about halfway along, it's all kinked to heck, and you're like, oh man, you got to go back and unwind it. That's what top isomerase does. All right, let's go to here first, and then I'm going to jump down to the other strand, because this happens on both strands. They just don't show it. The first thing that's laid down is a little tiny piece of RNA. It's called RNA primer, and this is really interesting, especially, again, in Bio 2. We think that the first genetic molecule on Earth was not DNA. We think it was actually RNA, which we'll talk about what RNA does in the next couple of chapters. But it does have a job here, even in the copying of DNA. So the first thing that happens is this enzyme called primary, lays down a little piece of RNA primer. So the bases of RNA, so this in case would be a ribose sugar, not a deoxyribose sugar, and there'd be uracils where there would be thymine. So that's just the primer, okay? So now, from here, what I'd like to do is jump down to here. All right, now there is a rule, and this is where the 3 and the 5 prime come in. When you read a piece of DNA, when you copy a piece of DNA, you must copy it. This is on the original strand, the one I just pointed to, in a certain direction. You must copy it from the 3 prime to the 5 prime end. You got to read left to right on that bottom strand. I'll get to the top strand in just a second. There's a lot going on up there, but let's make it easy for ourselves and stick with the bottom strand. So once that little piece of RNA primer has been put down, was it 3 or 4 bases long right here, then along comes an enzyme. And that enzyme, as many enzymes do, tells us what it does, and it's known as DNA polymerase 3. Now, you'll get introduced to 1 and 2 in just a second, but this is 3. And as soon as that little bit of primer is laid down, DNA polymerase 3, the primer sets the stage. That enzyme attaches, it would actually attach up here, to that original strand, and in the 3 to the 5 prime direction, so in the direction going left to right, it adds bases. So when it comes across a thymine, it attaches a loose adenine. It comes across a guanine, it puts a cytosine. 3 cytosines, it's going to put 3 guanines. It's going to do that. It's the tracing part, right? That part that up here, the DNA polymerase, as it moves along, is tracing the original in the green and adding the new bases following Chargaff's rule in the yellow. Meanwhile, the DNA molecule is being copied. Very cool, right? There's even a little sliding clamp, and what that sliding clamp does, it holds that polymerase. It's very machinery-like, man. That's what freaks me out. It actually holds that DNA polymerase in place. The single-stranding proteins up here, they prevent the DNA from wanting to stick back together before the DNA polymerase gets there. See, helicase splits it apart. Those hydrogen bonds are hydrogen bonds. They're like, they want to go back together, so the proteins get in the way and block it so DNA polymerase can read it. You know, so you don't want to have a situation where it unzips it. That comes back, you know, back and forth. That makes no sense. Okay, everybody, do you understand how that works on the leading strand? Very simply, we start with RNA primer down there on the left, our DNA polymerase on the sliding, DNA polymerase 3 in the sliding clamp, move down, making copies. Okay, here's where this whole idea behind the way chemistry is and the fact that you have to do things in a certain way. If you look at the other strand, the original strand, all right, the original strand, wait a minute here. Hold on 1 second, folks. 3 and 5. This is not the original strand. These are numbered wrong. Ooh, hold on 1 second, folks. This is a new picture of your book. First time I used it, I didn't look at. Let me make sure that 3 to 5, 3 to 5. Because you can't read. That's supposed to be 5. This is supposed to be 3. I think that's backwards. Yes, it is. Let me erase that. Those numbers need to be swapped. My notes. Let me write myself a note real quick. Hold on, folks. Just I need to do that. I never noticed that. This is the first time I've used that picture. I just noticed that. Swap ends. Yeah, but here's the problem on the other side, and this is why this is backwards. You understand what I'm saying, Ryan? That this should be 5. Hopefully I'm not screwing myself up later on. I have to change everything. This should be 3. Okay? All right, and the reason why that is if you look at the original strand is this one right here. The reason why I think those numbers are backwards, and I'm pretty sure they are, is you always have to read in the 3 to 5 direction. You notice that was not a problem with the original strand down here, okay? But the original strand up here is, remember, it's anti-parallel, so you don't start at the 5' end. You can't do with the RNA primer down here, right? You can't add RNA primer, slap on some DNA polymerase 3, a clamp, and head off down the road. It doesn't work that way. You're driving the wrong way down the road, okay? So here's how the other strand, it's called the lagging strand, because it takes a little more time. What it does is it, as the helicase unzips it, it adds the RNA primer, but at the other end. See? The RNA primer for the leading strand would be attached down here, and then we'd be off to the races. But because of the other strand, you got to go 3 to 5. You got to go to the other opened end and read back the other way. Okay? And so what happens there is the primase lays down a little bit of RNA primer, and then the ay. It's not the way it works. That picture is misleading. Crap. Sorry. I have another picture available. All right. Let me see if I can wing this a little bit for you. If I could draw it up here, what should be happening, and this is just the lagging strand. All right, so this is the lagging strand, and we know that we can't read from 5 to 3. Or you have to read from 3 to 5. All right, so what happens is the well, okay, anyway, what happens is the RNA primer is going to be this is just befuddling the heck out of me for a second. Give me 1 second, folks. I promise I will get this straight. I'm just trying to orient myself with the new picture. The idea behind the lagging strand is because you have to read in the other direction, you read, okay, you read a piece of the DNA and then you jump back and you read a piece before you. All right, so all of this has started down here. We've added a piece of RNA primer, and that piece of RNA primer has oh, okay. Geez. Should have had a picture before this. All right, so the RNA primer in the primase does start down here. Right, so it starts down there, and it's reading the original strand in the right direction, 3 to 5. All right, so it goes 3 to 5, 3 to 5, 3 to 5. Okay, and then no. Okay. All right. I'm gonna have to go back with you guys and get a better picture of this, because I can't do this with this picture because I think it's got a mistake. I will get back with this when we get back in lecture on Monday and go over this again with you. It's only one slide. The take home, at least for now, that you can come up with is when you copy DNA, one strand gets copied faster than the other. There's the leading strand and the lagging strand. But this picture is really throwing me for a loop as to how they're trying to illustrate that. So let me clear up my head, and I'll go over this again in class. Sorry about that, folks. I just looked at that, and for some reason, I'm just getting, you know how that vapor lock, like I used to do in physics class all the time. All right, let's move on there. As we're making copies of DNA and all that sort of stuff, things don't always work right? There's got to be some way to proofread or got to repair the DNA. Sometimes the bases don't match up right. You know, the A to a T and G to a C, that's not always correct. So there are enzymes that move along, kind of like those folks that move along with the hand cars on a train, I always thought about. And they try to fix these things. They find these little mistakes, and when they do, they call in an enzyme called nuclease, and we say, hey, nuclease, you need to cut out this. This is wrong. And nuclease comes in and does the chopping, cut. And then a DNA polymerase, that same one, DNA polymerase comes in and starts adding bases, the ones in the light blue, and fixes the mistake. Ligase means to stick together. Okay, so ligase means once you've added that new little part, think about DNA polymerase is filling a pothole, and DNA ligase is smoothing it over. It's going to add those bases, it's going to attach those bases to the other existing ones. Now, in Florida, of course, we have a concern, obviously, about skin cancer or melanoma, and what that is caused by that is ultraviolet rays, which are very powerful, will get sometimes adjacent thymines in your DNA to stick together. They're called thymine dimers. Dimers is referring to two. So you can see in the picture that the two thymines aren't sticking to the adenines, they stick to each other, and that can lead to melanomas. That can lead to misreading in the DNA, obviously, and problems, and obviously protein construction. Now, if a thymine dimer were to happen in, again, that part of your DNA that's not red, then it's no big deal. But if it's a part of the DNA that is red, that is going to be a big deal. And laying out in the sun, you don't necessarily know when that or how that's going to happen. Okay, so this is more lagging and leading strand, but again, I'll get to straighten out the mechanics with that picture in just a second, but or later on in class. But here's what happens. Because there is a lagging and a leading strand, on the lagging strand, the last piece of DNA that gets added is a primer, an RNA primer, all right? And so then there's another molecule that comes along that actually gets rid of that. It's another DNA polymerase, and it gets rid of that primer molecule, that little part in the red. And so what it does is every time you copy DNA, the copy that you have ends up with a jagged edge like this. You see, the lagging strand isn't in the right direction. This is the 3 to 5 direction. Now we got to be going this way, and the RNA polymerase is going to be set down, or the RNA primer is going to be set down, but that's not DNA. Later on in the replication process, something comes and cuts that out. Now, for whatever reason, it doesn't add anything back in. We get a jagged edge chromosome, and so the next time replication occurs, this piece doesn't get copied, and the chromosome gets shorter. So the next thing happens again with the RNA polymerase, the next round of complication, and the chromosome, this doesn't get copied, and the chromosome gets shorter. Through time, you get shorter and shorter and shorter molecules. Now, if you can see that on this X-ray fluoroscopy, the little yellow parts, those are the tip of the chromosomes. Now, if you're thinking, oh my goodness, every time my chromosomes replicate, pieces of it are being destroyed, they are, but don't worry about it. These things that I've labeled telomeres, telomeres are kind of like the plastic thing at the end of your shoelace. What's that thing do? it has a name for it. It starts with an A, which I never remember. But what's that little plastic thing in the end of your shoelaces do? Because if you're like me, sometimes the plastic thing wears off, and then you know what your shoelaces do. And when I was a kid, they always wore off. Was it all Velcro shoes now? You guys have shoelaces. Yeah, so if that plastic thing fell off, the string would unravel. Think about these telomeres as being the end of DNA molecules that are the plastic part of the shoestring. All right, what I mean by that as well is that's part of the DNA that's not read, not used. It's at the end of the DNA. It's like packaging material. It's not. So the fact that every time you replicate, every time you lose a piece of it is no big deal initially when you're young. You got a big chunk of that. You got a big shoelace protector. As you get older, and every time you copy a piece of DNA, because of what you see right here, your chromosomes get shorter. The tips do. The tips get shorter and shorter and shorter. Now, here's what happens when you get really old. You run out of telomeres. Now you get to the part where that little plastic thing is completely gone, and the strings of your shoelace begin to unravel. So one of the research that's being done is to aging, what causes aging. I mean, we know things fall apart in entropy. That's just physics. But maybe one of the ways to slow down aging or control aging is examining these telomeres. See, basically the idea is if you could stop this from happening, then the chromosomes would not shorten eventually to a point where they start, destroying good genetic material. Right? If the chromosome gets short enough, then you start cutting into the stuff that really is genes, and that causes problems. And we think that might be part of the aging process. Telomeres. Invent a way to prevent telomeres from occurring. Invent a way to prevent molecules from getting shorter like this, and you'll be a rich person. Because, again, entropy being what it is, I think we would all have to die at some time, but this would prolong it quite a bit. All right, let's talk about some mutations to DNA and their effects. On the left-hand side, I'm talking about what would happen if the wrong nucleotide was substituted. So in other words, I'm changing the letter. Up at the top where it says wild type. Wild type in genetics means the way the DNA is supposed to be, if you will, the normal way. And as I've mentioned before, the DNA is a code. Now, down below is the sequence of amino acids for a particular protein. I'll talk in subsequent chapters how the code gets turned into those amino acids, but you ought to recognize that what I'm showing you down there, right here, is the primary level of protein structure, right? The order of the amino acids. So it's a very small protein, only 4 amino acids long. And all that is coded for in the DNA in ways I'll show you later on. Okay, so let's monkey with the code. Let's change it. So instead of this, where are we, right here? Instead of this guanine I just circled up at the top, an adenine gets inserted. So an adenine gets inserted, so now it's a thymine on the other side. All right, well, when you look at the genetic code, which we'll do later on, changing that last C right here, because that's where the code is going to be read on the messenger RNA, has no effect. You still get the same amino acid. If you get the same amino acid, the amino acid still folds the same way. Do you have a mutation? Yes. Do you have a mutation to the junk DNA? No. You have a mutation to your actual DNA that's read. Does it have an effect? No, it doesn't have an effect. You get the same amino acid. Does not matter. We call that a silent mutation. So silent mutations could be ones to the junk DNA, or they could be ones that cause no effect. No problem, right? It's a different letter, but it codes for the same amino acid. Now, again, we'll talk more about the coding later on. I'll emphasize again this to you. Now, what would happen if we change one of the bases, like in the second, where we do change the amino acid? So now instead of glycine, we have serine. Well, I don't know. It kind of depends on what serine does. It kind of depends on the protein. It kind of depends on if serine is, let's say, a hydrophobic amino acid and glycine was a hydrophilic, and I don't remember off the top of my head if they are one or the other, but that would cause the protein to fold differently. It might matter if serine had particular side groups, that without those side groups in the right places, hopefully this is all making some sort of sense to you guys, the protein didn't fold back on itself the right way. It might have no effect. It might be kind of like a silent mutation, but the effect on the protein is so minimal that remember, like for an amino acid, an enzyme, has an active site with a particular shape. Well, what if that shape was just a little bit off because it was a serine? You remember enzymes have induced fit. They kind of accommodate minor changes in shapes. Could be big deal on a missense, right? Could be, for example, sickle cell anemia is caused by a missense. One base different causes that hemoglobin protein problems that we talked about early on, right? Down below a nonsense. If something happens where the group of letters right after that first amino acid tells the cell to stop, some of these letters code and say, okay, that's the protein, stop. That's the end of the protein. It's kind of like using one of those label makers, dun, 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 then you cut it off. Well, if something were to happen to mutate one of those letters so that the machinery of the cell says, okay, first amino acid is methionine, stop. Well, in that case, it's nonsense. You make nothing. Methionine is an amino acid, not a protein. So if a mutation occurred that went methionine, stop, you don't get that protein at all. Say maybe being lactose intolerant is because you don't have the lactase enzymes. That could be a reason why. Maybe that code there says to start with the first amino acid, stop with the next one because of some mutation, so you don't get anything built. Now, the other ones on the other side are going to be talking about nucleotide pair insertions or deletions. So instead of a substitution, we're going to do something that screws up the order in which these are read. You see, and again, we haven't talked about this yet, but we will. But these letters down here are read in groups of 3. And so if you insert, like that adenine that's being inserted, you shift the frame because you read 3, you read AUG. Now, in the wild type, you read AUG, AAG, I'm circling up above, but now you're reading UAA, because you go AUG, UAA. Well, that's a stop codon, because you added that extra A in there, caused it to be a stop codon. You get a start, you get methionine, stop, you get a nonsense, but it's called a frame shift mutation. If you take one out in the second one, you get a frame shift mutation. You read 3 at a time. AUG, that's methionine. AAG, that's lysine. The next 3 should have been UUU, which is phenylalanine, but the third U is missing. Does the DNA machinery know that? No, not at all. So when it goes to UU, it just picks up the next base, and it picks up G. Now, that doesn't code for phenylalanine. That codes for leucine, and then that screws up everything down the road. See, a frame shift mutation like that is going to have big effects, because a piece of DNA that's being read, or in this case the RNA, can actually code for many, many, many proteins. If you had a frame shift mutation at the beginning, whoa, is everything downstream going to be screwed up? It's kind of like misbuttoning a button shirt. You know, if you misbutton that first button, you get to the bottom, you got to unbutton the whole thing. Everything's messed up. There are also times in which an entire group of 3 letters might be missing, like part of the DNA mutates or something happens in which that group of 3 letters is missing. So this group of 3 letters that does the coding, you completely miss an amino acid. So instead of methionine and lysine, you go methionine and then phenylalanine in this case. Now again, what kind of damage does that cause? That's going to depend on the protein, the amino acid, the amount of change. Remember, amino acids, we separated in those that were acidic and basic, hydrophilic and hydrophobic. If a mutation occurs and an acidic is swapped for an acidic, or hydrophobic for a hydrophobic, maybe minor change, maybe big change, maybe minor change. But if you're swapping like an acidic one for a basic one or a hydrophilic one for a hydrophobic one, then you might expect to have maybe a bigger change. And of course, not to mention all the side groups that you change. Remember, the tertiary level of protein structure was when all those side groups came together and how that bonds. Okay, that's all I have for lecture. Like I said, I'm going to go back and take a look at that image of the leading and the lagging strands again. I might replace it, but that's the first thing I'm going to go over. Oh, shoot. Monday we have an exam. Don't we? Wait a minute. We have an exam. We have no lab on Monday. Okay, good. So we're not going to have we'll have a lecture exam Monday, and then when that's over, we're not going to have a lab, but I'm going to stick you guys around for a little bit to go over that I want to go over, And also I want to work a little bit more with you guys on the genetics problems and getting you guys ready for Lab Practical 2 as well, which is the genetics problems in mitosis. So I want to give you guys some time in the lab to review the slides and all that kind of stuff. It won't be a long lab, obviously, just like I said, a little bit of lecture and then a little bit of review. Well, as much review as you want, actually. So don't forget your exam is Monday, chapters 7 and 8. We don't do 9, chapters 10 and 11. And don't forget any assignments that are due in that chapter 2 as well. Okay, that's all I got for today. I'm going to go ahead and stop the recording. Got any questions or anything? More than welcome to stick around. If not, have a good day, Ryan. You're welcome.